So, in our last two discussions, we looked at what are the ways in which organisms generate variation both in the long time scale in terms of creating new genes or in the short time scale in terms of phenotypic changes or cryptic genetic variation and so on. And I told you that all these changes, they leave their mark on the genome of a species, and therefore, when you look at the genomes, they act like books which contain the tales of all the previous generations. So, what exactly are the stories that they tell us, and that is what we are going to look at in this discussion, in today's discussion. What are the kinds of patterns that you can see in genomes which allows you to infer various things about how they have evolved. But before we get on with that, there is a certain concept that we need to get cleared, and that is related to mutations. So, remember that when you have mutations that are leading to substitution of a base, transition or transversion, then there are two kinds of mutations that are possible. One, what are known as non-synonymous mutations, where because of the mutation the amino acid that is incorporated into the protein changes. So, these are denoted as N, and the other are the so called synonymous substitutions, wherein there is a mutation, you can see it in the gene, but it does not lead to a change in the protein structure. Now, why do synonymous mutations happen? Because we have about 64 codons, 3 are stop codons, so about 61 amino acid coding codons, but we have only about 20 amino acids, which means that there is a redundancy for many of the amino acids, which means that if you end up changing one of the base pairs, then it is entirely possible that you get to the same another version of the codon which codes for the same amino acid, which is why synonymous substitutions happen. Now, suppose you have two genes or the same gene from two different species, okay, and now you are trying to figure out that what is the difference between these two genes in terms of the sequences. So, when you are trying to compare the sequence of a gene from two species, then you end up calculating two quantities. Okay, you essentially need to see how many non-synonymous substitutions are there and how many synonymous substitutions are there. But the number of ways in which a non-synonymous substitution can happen is much greater than the number of ways in which a synonymous substitution can happen given an open reading frame. Therefore, what you need to do is you need to scale the number of substitutions that you are seeing by the total number of ways in which those substitutions can happen. And if you do that, then you get two quantities, so called dn and ds. So, when you take the number of non-synonymous mutations and divide them by all the number of sites in that gene where a change would lead to a non-synonymous mutation, then that ratio, that scale value is what is called as dn. And similarly, when you do the same thing for synonymous mutations, okay, how many changes have actually happened divided by in that gene how many changes would lead to synonymous mutations. Okay, so that is the thing. So, that stuff is known as ds. Okay. Now, we are going to look at the various patterns that one sees, and the first prior to that, why are you calculating dn and ds? That is because the ratio of dn to ds actually gives you a very nice insight into the nature of selection that is happening on a gene. So, suppose you have a situation where between two species you see your dn ds is less than 1. I am sorry, for a gene you see your dn ds is less than 1. Now, what will that mean? That means that there is a purifying selection or negative selection happening. Why? Because dn ds is less than 1, which means that non-synonymous mutations are happening much less compared to synonymous mutations, which means that anything that is leading to a change in the protein structure is typically being not favored by selection, which typically implies that it is an important protein, it is a conserved protein, and that is why the selection on that is of the negative type. Any departure from the existing version is not easily tolerated. So, this is the kind of stuff that you see in, for example, housekeeping genes or ribosomal genes, right. I mean, there are many housekeeping genes, ribosomal genes are one example of that. Now, suppose you now see DNDS is approximately equal to 1. What does that mean? That means that synonymous and non-synonymous mutations are roughly happening at the same rate. What does that mean? That means that as far as the gene sequence is concerned, it does not really matter whether you have synonymous or non-synonymous mutations, and essentially mutations are just happening randomly, and there is nothing which is altering the fixation rate of those mutations. And when that happens, then you typically infer that this is a unimportant gene, or more likely that it is a gene which is not even leading to a protein. So, based on our previous examples, our discussion, you know that dead genes that are not producing proteins, they are known as pseudogenes, right. So, pseudogenes typically will end up showing this property. The third possibility, I mean less than 1 equal to 1, then the only other possibility is DNDS is greater than 1. So, this is the situation where non-synonymous mutations are accumulating at a faster rate than synonymous mutations, which means that this is a gene which is under very strong positive selection, and that probably means that this is increasing the adaptation of the organism, and in that sense it is very continuous evolution of this gene is very important for the fitness of the organism. So, for example, this is the kind of stuff that happens in all those genes which are allowing the organisms to evolve and adapt to new challenges. So, for example, you let us say have a bacteria, and you are hitting it with different kinds of antibiotic in a hospital situation. So, there are all kinds of genes which are responsible for antibiotic resistance, and those genes are continuously being strongly selected for. So, any positive non-synonymous mutation, any non-synonymous mutation which is leading to positive fitness effect is going to be beneficial under that situation, and therefore, the total number of non-synonymous mutations is going to be greater than the number of synonymous mutations. So, these values, they quickly tell us what is the kind of selection that is happening on the organism, or rather on that gene. Now, based on this, now we can look at our first property, which is that mutations accumulate at a roughly constant rate. What do I mean by that? So, here I am showing you a graph wherein the x-axis is showing the time in millions of years since the most recent common ancestor of human and 9 other species, numbered 1 to 9 on this blue line, the time since they diverged. So, what they did was they, let us say, okay, let me show you what the species are. So, let us say one is chimpanzee, which is over here. So, what this is telling you is that humans and chimpanzees, based on their fossil data, they diverged somewhere about a few less than 10 million years ago. Similarly, number 7 is chicken. So, the ancestor of humans and chickens, they diverged somewhere about say 320, 30 million years ago, and so on, right. Now, that is on the x-axis. On the y-axis, you have the number of various kinds of substitutions per site between humans and those species, and this is not based on 1 or 2 genes, this is based on about 4,200 loci, right. Now, there are 3 kinds of substitutions that are being talked about over here. The blue one, this line, is the synonymous substitutions. The red one, this one, is amino acid substitutions, and DN, this one, is non-synonymous substitutions. Okay. And of course, the corresponding lines, the blue, the red and the green, these are the regression lines. Now, what do you observe? The first and the most interesting thing that you observe is that there is a strong correlation between when the species diverged and how much sequence difference is between them. In other words, you can see that this is almost linearly increasing. Okay, in the context of synonymous, it is slightly, ever so slightly noisy, but in the context of the amino acid substitutions and the non-synonymous substitutions, it's very strongly linear. In other words, the rate at which the mutation is accumulating, that is almost constant. And the second thing that you see is that if you look at these three classes, synonymous, non-synonymous and amino acid, the rates are different, right. I mean, you can straight away see that synonymous substitutions are accumulating at a much faster rate, and in this particular case this is about 5 times faster compared to the non-synonymous ones, the green ones here. Okay. But within a particular class, that thing is still changing linearly, which means that the rate is constant. Now, obviously, if you have an observation like this, there is a very important practical consequence of this. What is that? The practical consequence is that you are seeing that at least some genes, they accumulate mutations at extremely constant rates, and all those genes which do that, they can be used as molecular clocks to estimate the time of divergence of species that for which we do not have any fossils. Okay. Now, how do you do that? So, just to show you what I mean. So, suppose you have two species and between which you want to see what is the time to divergence. So, what you do is that you compute the number of differences in amino acid sequences for proteins or nucleic acid sequences for genes for the given pairs of species. Okay. Say humans and some snakes. Now, why did I say snakes? Because if I look at this particular diagram, I do not have snakes in this data. Okay. But now, what we can do is, suppose you have a calibrated diagram like this, where you already know that the relationship between divergence time as determined from fossil data and the substitutions per site, the lines look like this. And what you can do is, you can take this calibrated relationship where both fossil data for divergence and substitution rates are available, and then use that for your unknown species for which you do not have fossil data. So, for example, let us say that we observe that our synonymous for the snake that we have in mind, the substitution rate for that is, let us say, somewhere over here. Okay. So, from this data, what we can do is, now we can go down and say that the ancestor of that snake and humans, they diverge from each other, say somewhere about 250 years ago, right. In other words, even if you do not have the fossil for that snake, given this relationship and given if you can compute this substitution rate, you can figure out how long back was the divergence. And this concept is actually used extensively in molecular phylogeny to estimate the divergence time between species, because obviously fossil records are very sparse. You do not always get a nice fossil to say that okay, this is the time at which snakes diverged from whatever the ancestors of humans. So, this ends up becoming very useful, but this leads to a massive question. Why should sequences evolve at such constant rates? I mean, we know that mutations themselves are not always we saw that they end up having very different rates, transition rates are different from transversion rates and so on and so forth. But if all that be the case, then why should, at the level of the genome, why should it look as if that they are evolving at a constant rate? So, the answer to this question came from the work of the famous Japanese molecular evolutionary biologist Moto Kimura, and he proposed what is known as the neutral theory of molecular evolution. So, what exactly did Kimura say? So, he said that mutations that lead to beneficial or harmful fitness effects, they have either already been fixed or been eliminated. Okay. Now, then he says that such mutations, beneficial or harmful, they actually have not contributed significantly to the variation of the existing sequences. All right. Now, prima facie, this will look like a very bold assumption, but if you think about it closely, it is not. So, if you remember when we were discussing the distribution of fitness effects, DFEs, we said that most of the mutations, they are actually not going to have any effect on fitness, but amongst the mutations which will have an effect on fitness, a large fraction of them are actually going to be deleterious. So, all these deleterious mutations, since they are deleterious, what will happen? Over time, long time, there is going to be selection against those mutations and they are just going to disappear from the population. At the same time, those mutations which are beneficial, if the beneficial the benefit is reasonable, they are going to slowly accumulate and at some point they are going to get fixed. So, that stuff is not going to lead to any variation at the level of the population, because all individuals will have it, all the harmful mutations are anyway getting selected out. So, when you think about variation that is there inside the population, that actually is going to be contributed primarily by those variations which do not have any effect on fitness, essentially those mutations which are neutral, right. Now, we already saw that if there is something which is neutral, there is no selection acting on it, then the primary thing that will lead to the changes in their frequency in the population is going to be random genetic drift, right. So, Moto Kimura essentially ended up saying that, look, at the level of the molecule, at the level of the sequence, the primary force that is leading to the variation that we see around us, that is actually random genetic drift and not selection. Now, this of course led to a massive debate, the so called selectionist versus neutral mutation debate. But over time, people essentially figured out that Kimura is not really trying to say that selection does not work. He is essentially saying that at the level of the sequence, it is the neutral mutations which play a greater role, and as we went forward we realized that sometimes there are situations under which that works, there are other situations under which they do not work, and all those things we are going to see in the context of today's discussion. But the main thing that I want you to take from this is the observation that random genetic drift of selectively neutral mutant alleles have led to the majority of the intra and interspecific variation. This is what neutral theory of evolution tells us. Now, if this be the case, we already know that, or rather we know that the magnitude of drift depends on the effective population size, right. Therefore, the amount of genetic variation within a species is going to be proportional to its effective population size. Why is that so? Think about it. We have seen that when the effective population size is very low, the effect of drift is going to be very high. And when the effect of drift is very high, we know that there are only two things possible. Either a particular mutation or a particular allele is going to get lost, or it is going to get fixed, right. Frequency equal to 0 or frequency equal to 1, and once these happen, barring back mutation, that is going to be the stable state. So, obviously, the smaller the effective size, the lower is going to be the genetic variation that is present due to neutral alleles in the population, which is the same way of saying that or another way of saying that if the population size or the effective population size is high, that is when the amount of genetic variation in the population will be higher. The next major thing that neutral theory claims, and Kimura actually shows this using mathematical derivation, we are not going there, but he claims that the rate of neutral mutations is going to be constant over time, and therefore, the rate at which these neutral mutations become fixed in a population, that also becomes constant. So, that is the derivation that I am talking about. Now, think about it. If the rate of neutral mutations in a population is constant and the rate at which these neutral mutations are getting fixed is also a constant, then taken together, what is going to happen? That means that for these any two species, the rate at which these mutations will keep on accumulating will become a constant over time, which will show up in the kind of figure that we saw in the previous slide, where as time progresses, the divergence time progresses, the amount of mutation accumulating goes up linearly at a constant rate. Okay. So, this is the explanation for why you have molecular clocks. Now, that was the first observation. The second observation, very interesting, is that the different parts of the genome actually evolve at different rates. Now, what do I mean by that? So, here is some figure about the evolutionary rate for different components of the genome. So, this is estimated from differences between humans and chimpanzees, and these are measured as the number of differences per nucleotide site multiplied by 10 to the power 3. So, that is what is there on x axis, that is what we are calling as evolutionary rate. Now, we have 4 different kinds of things here. These are the pseudogenes, and you can see that the rate at which they are accumulating differences is the highest. These are coding genes. You can see that the rate at which they are accumulating mutation is the lowest. Now, in some sense, both these are obvious. Why? Because in pseudogenes no selection is happening. So, there is all mutations are selectively neutral over there. So, it will keep on accumulating mutation without any cost to the organism. So, obviously, evolutionary rate of divergence over there is the highest. Similarly, coding stuff, non-synonymous mutations, many of them will end up affecting the fitness, and therefore, the rate at which it will accumulate non-synonymous mutations in the coding gene is going to be much less. So, these two are taken care of, these two are understandable. Why are we getting this scenario where intergenic regions, that is if you have 2 genes, there are nucleotide sequence between them, that is what is known as the intergenic regions, and then you have introns. What are introns? So, you know that you have eukaryotes, you have these in the genes, you have parts of the gene which are known as exons and the parts of the genes which are known as introns. So, the exon, intron, that is the structure of the gene. And when the mRNA is formed, then the entire thing is transcribed, but after that happens, the intron portions are cut out and all the exons joined together, and it is this edited version of the mRNA which goes for translation. Now, obviously, if there are changes that are happening at the intron level, or if there are changes that are happening in the intragenic regions, then they should be selectively neutral. In other words, in them you should have the evolutionary rate, rate at which mutations are accumulating, that should be equal to that of the pseudogenes. But you are not really finding that. You are finding that these two things are actually intermediate between the pseudogenes and the coding genes. Why? That is because we now know that both the intergenic regions and the introns, they can actually affect the fitness of an organism. Why? Because the introns, they play a role in gene regulation, and why is that? Because we know that there is something known as alternative splicing. So, depending on which introns are there, the same version of the mRNA can lead to multiple proteins through different combinations of the exons, and which exons are getting combined with each other, that the introns play a role in determining that. And secondly, the introns, it has also been shown that they play a role in determining RNA stability. That is why if there are mutations happening in the introns, they are not completely selectively neutral. Secondly, if you look at the intergenic regions, they do not have the coding region for sure, but they have regulatory sequences, they have promoters, they have enhancers, and what we now know is they have many non-coded RNAs. We talked about these piRNAs, or you have what are known as siRNAs and so on, and all these things, they play a role in terms of determining the fitness of the organism. They affect the organism's function, and that is why if you have mutations in these regions, then sometimes they can affect the fitness of the organism depending on where they are happening. But obviously, they are not as critical, as crucial, I'm sorry, I'll take that back. If there is a mutation that happens in a regulatory sequence or a promoter sequence, it is extremely crucial. But what I'm trying to say is that not the entire stretch of intergenic region or not the entire stretch of the intron is as crucial as, say, the entire stretch of the coding region, which is why the mutations in these regions, intergenic regions and introns, they accumulate at a rate which is intermediate between the pseudogenes and the coding regions. Great. So, also different sites in the genome, they will evolve at different rates. Now, how is this different from what I just now told you? So, what I just now told you is in terms of the different kinds of regions, pseudogenes, coding regions and so on. What I am now telling you is in terms of positions in the open reading frame. So, you know that we have codons which are made of 3 base pairs, right, but the 3 base pairs, they do not accumulate the mutations at the same rate. So, if you look at it, so here what we are showing you is the proportions of base pairs that differ in the DNA sequences in the mitochondrial genes called COI between pairs of vertebrate species. So, multiple pairs have been taken, and this is plotted against the time since their most recent common ancestor diverged, and that is why this is in terms of millions of years. Okay. So, they have taken pairs of species for which the fossil data is available. So, based on that fossil data availability, they have figured out what is the time duration between their divergence. That is what is there between that is what is there on the x axis, and then on the y axis they have plotted what the sequence divergence in terms of the third base pair in the codon, the second base pair in the codon and the first base pair in the codon. And what you straight away see is that when it comes to the third base pair, then mutations over there accumulate extremely fast, but then after some point they more or less stabilize. Okay. Whereas, in terms of the first and the second base pairs, the mutation accumulates much slower, and particularly for the second base pair, it accumulates really slowly. Now, why is that so? Remember the wobble hypothesis. So, we know that all the 3 base pairs in a codon are not equally important. So, most of the cases, the redundancies that you have in terms of the genetic code is because of the wobble in the third base pair. So, if you end up changing the third base pair, then in many cases the changes are going to be synonymous, which is why it is very easy for a mutation over there to be selectively neutral, and anything that is going to be selectively neutral, as we know, is going to get accumulated much faster. Now, of course, there is a limit to this. Okay. There is only so much wobble that is there at the third base pair. So, that is why after some time, when that in terms of divergence, after some time there is no further increase in terms of divergence at the third base pair. However, when it comes to the first and the second, then the redundancy over there is much less. In the context of the second, there is hardly any redundancy. So, because of which, if there are mutations over there in the first or the second base pair, then most of the time they are non-synonymous, and that is why they are not selectively neutral, and therefore, they do not end up accumulating as much as what happens at the third base pair. So, in that sense, this is again congruent with the fact that anything that is selectively neutral will typically accumulate much faster. Now, this brings us to a very important relationship between the genome size and the complexity of the organism. Now, let me start by saying that there is no accepted definition of biological complexity. In fact, there is no accepted definition of complexity to begin with. But for practical purposes, roughly you can think of those organisms which have, let us say, more types of cells, or which have more kinds of tissues, or let us say organisms which have greater division of labor for various functions. Okay. those are the ones which are more complex compared to those organisms which let us say are lesser along these axes. So, for example, think of a E. coli. The E. coli does everything on its own. The single cell of E. coli, it has to find its own food, it has to reproduce, it has to, if there is a danger, it has to go away from it. Everything has to be done by that one cell. However, think of an organism like a human. Okay, I use a different set of cells for seeing, a different set of cells for hearing, a different set of cells for reproducing, and so on and so forth. So it is in this context we have more number of cells, we have more number of tissues, and there is division of labor across those cell and tissue types, and therefore we can reasonably say that a human body is more complex than a E. coli. Of course, there are philosophical arguments that one can make, but let us for the moment assume that that is how we think about complexity. So, fossil and DNA evidence tells us that in general, this is very important, not always, but in general, evolution has led from simpler forms to more complex forms. Okay. So, that is why the earlier forms, the ancestral forms were typically single cellular, then at some point multicellularity evolved. The ancient multicellular organisms, they had very few kinds of cells, but then the number of kinds of cells started growing and so on and so forth. So, we have, of course, there are situations like say parasitism, where from complexity one has gone towards simplicity, or an organism has ended up losing its function, ended up losing its various tissue types and so on. But in general, it is from more simple to more complex forms. But mechanistically speaking, where is this increased complexity going to be coded? Okay, how are you going to get the features that are needed for this greater complexity? And as we have been talking about that, whenever you need novel traits, then the place from which they will come from at some level will have to be some genes. Okay, you can do certain things by changing gene expression levels, but beyond a point you need new genes, right? Therefore, one can reasonably argue that as you go from more simple to more complex forms, the number of genes that has to increase. In other words, we expect to see a positive relationship between the genome size and the complexity of a species. So, this is the expectation. Does it really happen that way? So, what I am showing you over here are the ranges of the genome size for a bunch of organisms. So, we have the genome size on x axis and we have different kinds of organisms on the y axis, and as you can see, these are the ranges, right. Now, of course, you can see that for any class of organisms there is a huge variation which is not entirely obvious. So, for example, all prokaryotes are supposed to be single cellular. So, why do I expect this much of a variation? Okay, maybe. But what ends up becoming more interesting is how, if you look at the overlaps. So, for example, here right at the bottom we have the birds, the mammals and reptiles, and you can see that the range in their genome size is somewhere about one order of magnitude between 10 to the power 3 to 10 to the power 4. But now look at, for example, algae, or look at protists, these are the best. Okay. Look at the amount of variation that you have in protists, and this entire zone is actually greater than this stuff, than all birds, mammals, reptiles, suggesting that there are quite a few protists who have genomes which are much larger, in orders of magnitude larger. Remember, the x axis is in the log scale. So, which have orders of magnitude larger genomes compared to birds, mammals and reptiles. Similarly, look at for example, this fungi, all right. Again, look at the amount of variation that they have over here, but look at the protists. These are far, these protists are single cell organisms, but they are far outscoring the fungi. Similarly, many vascular plants, Okay. Even the simple Arabidopsis and all, many of these guys have genomes far larger than mammals and birds and fishes and chordates, whereas as far as we can see, the they are not as complex, they are much more simple. So, what exactly is going on? Now, here I have to introduce a term, and that term is C value. What is it? So, the amount of DNA that is present in the haploid phase of the life of that organism, that is what is known as C value. Now, why do we need to think about the C value? Because some organisms are haploid, some organisms are diploid. So, you can't really do a apple to oranges comparison. So, in order to do a proper comparison, you bring everything down to the haploid complement. Okay. So, when I was talking about it over here, this is all about the haploid genome content. So, this observation that there does not seem to be a relationship between the C value of a species and its complexity, this is what is known as the C value paradox. Genome size typically does not correlate well with complexity. Okay, and this is our fourth pattern. Okay. Now, the question is, why is that so? So, one potential solution to this comes out if you look at the composition of the genome. Okay. So, we are showing you the composition of the human genome over here. So, you see this little tiny sector over here, these are the coding sequences, the exons, and less than 2% of the human genome is actually devoted to protein coding sequences. So, what are the major things? So, turns out that the major things are actually different kinds of transposable elements, the jumping genes. About 50% of the total genome is composed of these transposons, and then about 25% are introns, which which is actually a little more than 25% to be frank. So, now how does this solve the C value paradox? Because we said that an organism's complexity should be related to its genome size, but implicit in that is the assumption that the whole of the genome is contributing to the organism's complexity. But if the whole of the genome is not contributing to the organism's complexity, as we can see over here, only 2%, less than 2% is doing so, then that probably means that that will explain why we are seeing this discrepancy. Maybe the organisms which actually have larger genomes, they are the ones who end up having a lot of quote unquote junk in their genome, which is not contributing to the complexity, which is why even if the genome size is large, complexity is small. Possible. But in order to understand whether that is indeed the thing or not, we have to ask the question that is this thing that we saw in the context of the human, is that true for all organisms? So, is it true that in all organisms the amount of non-coding DNA that we find is similar? So, that is the question that was asked in a paper by Michael Lynch et al. And not et al, Michael Lynch himself. So, this is a very interesting graph. So, this is showing us the relationship between a genome's total DNA content, the genome size, and the amount of its protein coding sequence. Okay. And what has been done is that there are 3 lines over here. So, this line is for 100%, which means that if a point is on this line, then for that organism 100% of its genome is coding sequence. Okay. This line over here is 10% coding sequence, and this line is over here is 1% coding sequence. Okay. So, note what is happening over here. So, if you look at this legend over here, you realize that these guys, these are the ones which have 100% coding sequence. Who are these? These are the bacteriophages. These are the eukaryotic DNA viruses. So, viruses which affect the eukaryotes, and these are the prokaryotes. Okay. these are the guys. So, these are the ones with one little exception here and there. These are the ones in which the coding sequences actually occupy 100% of the genome and they hardly have any non-coding DNA. But then the moment you come to, let us say, here, okay. So, here these are the animals and these are the land plants, you see that they are diverging away, which means that these are the organisms which have a lot of non-coding DNA in their genome, and unicellular eukaryotes, sorry, these are the ones which sit somewhere intermediate between these guys and the plants and the animals. So, this is telling us that there is a lot of variation in the genome size, as we can see over here, but it is only in the animals and the plants where you have a lot of the non-coding DNA. All these other organisms, bacteriophages, the DNA viruses, the prokaryotes, etc., all those which have to typically replicate extremely fast, okay, generation time is extremely small, those are the ones that do not have all this non-coding DNA business, whereas all those organisms which have relatively longer life cycle, those are the ones which have all the non-coding DNAs, and unicellular eukaryotes, which are somewhere in between in terms of life cycle, are also somewhere in between in terms of coding non-coding DNA fraction. So, this tells us that coding to non-coding ratio of genomes vary across taxa, with simpler organisms generally having more streamlined genomes. More streamlined means less amount of non-coding DNA. Great. But if we have found this, then that simply means that look, it is the non-coding DNA which is screwing up the calculation. So, let us forget about the non-coding DNA. Let us go back to our C-value paradox, and instead of saying that there should be a positive association between the genome size and complexity, let us simply say that there will be a positive association between the number of protein coding genes and the complexity of the organisms. Okay. Does this prediction hold? I will just show you a few examples. So, here we have some species, and I have organized this in ascending order of number of genes, based on whatever we know from their whole genome sequences. So, this is Drosophila melanogaster right at the bottom, this is Z elegans, this is a nematode, these are us humans, this is a zebrafish, this is Arabidopsis, this is Daphnia pulex. Okay. Now, look at their genomes. Drosophila has reasonably, among all these examples that I have taken, they have the smallest number of genes, but this is a nematode, okay, which is, for all as much as we can see, a far simpler organism than Drosophila melanogaster, yet it has about 6,000 extra genes. Homo sapiens, as humans, we have about 19,000 to 20,000 genes, which is roughly in the same zone as the C. elegans, which is a small tiny nematode. Zebrafishes, they have way more genes than us. Arabidopsis, the small plant, the thalcrase, very widely used in by plant molecular biologists, that damn thing has 27,000 genes. And Daphnia, that microscopic 4-5 millimeter long organism that we talked about in one of the previous lectures, that class, that thing has 31,000 genes, okay, about, roughly speaking, a little less than double compared to the humans. So, taken together, it is pretty obvious that there is no correlation between the number of protein coding genes and the complexity of the organism either, and this is what is known as the G-value paradox in literature. Okay, no association between the number of protein coding genes and the complexity. So, now obviously comes the question, why? Why do we expect the G-value paradox, or rather what explains the G-value paradox? So, turns out that quite a few mechanisms have been proposed to explain the G-value paradox, and I do not think there is one mechanism, all of these probably act simultaneously. So, for example, in many cases, organisms which have a lower genome, a lower number of genes, for example, as humans, we have more sophisticated controls on gene expression. So, for example, epigenetic mechanisms. So, that even if you have relatively fewer number of genes, if you have good epigenetic ways of turning them on and off and thereby controlling their spatial and temporal aspects of expression, that might lead to more diverse characters. Similarly, in many of these organisms, maybe the proteins, they are multifunctional. Instead of one protein doing just one job, maybe one protein is doing multiple jobs. Or maybe the proteins are such that the number of interactions that are happening in the metabolic pathways is much greater. Or maybe, I mean, this we already know, is there, alternative splicing and post-translational modifications. All these things are leading to the fact that you have just one gene, but the number of functional products that are coming out of that gene is much greater, which obviously can lead to greater complexity with a lower number of genes. And finally, and this is again something that has been extremely well shown, that in many of these organisms which have lower genome size, they have lots of non-coding RNAs, miRNAs, siRNAs, and all those other RNAs that I talked about. And all these things, not only do they provide greater control of gene expression, in some cases they might end up doing certain functions on their own, none of which will obviously show up in the gene count. So, the basic idea is that the complexity of the organism can be modulated even with a lower number of protein coding genes if you have all these other features. Great. So, till this point, and when I say till this point, I mean right from the first discussion that we did in week 1, everything that we have discussed till now about the various forces of evolution, selection, drift, mutation, migration, etc., and how these forces act with each other. Everything that we have discussed has been within the so-called modern synthesis or the extended evolutionary synthesis framework. And this framework presupposes that life has a reason, and then it starts talking about how things might have changed after that. Okay. But the crucial question of how exactly did life originate, that question, which is perhaps supposed to be the biggest question in the whole of biology, that question actually falls outside the purview of both MS and EES. Now, it is a very, very hard question. Why? Because there are no fossilized remains of those absolutely early ancestral life forms. Okay. Therefore, even if we end up having some nice theory about how those things might have worked, it is very difficult, close to impossible, to have proper evidences that okay, this is the way it happened and not the other 15 alternative hypotheses which have been proposed. So, it is a tough one, but it is something that is extremely interesting nonetheless. And once you understand that, then the other interesting thing that we have not worked on till now is about us humans. How exactly did we originate? From where? What are the various forces that have worked, evolutionary forces that have worked on us to bring us evolutionarily where we are right now? So, these are the kind of questions that we are going to discuss in our next discussion, which is going to happen next week. Okay. See you till then. Bye.